use glycolysis, right? They have a glycolytic pathway, and so a lot of the enzymes that are used in glycolysis are thought to be pretty ancient. They've been around for a long time. And so if we look at studying those enzymes in the glycolytic pathway, how they function, how they're regulated, the kinds of inhibitors that will inhibit their activities, a lot of the work that's been done studying those glycolytic enzymes in, say, bacteria or fungi like yeast turn out to be true for enzymes that are found in higher organisms that carry out glycolysis as well. So we think these are really very ancient pathways. And the other thing is that glycolysis doesn't involve oxygen, right, as an electron acceptor. And so the thinking is that this is likely to have been a pathway that evolved in cells that were living on the Earth at a time when there was very little oxygen available. So they had to survive without the availability of oxygen. So if looking in the fossil record, we can find evidence of bacteria going all the way back almost 3.5 billion years, but there's other evidence that suggests that oxygen was not actually abundant in the Earth's atmosphere until about 2.5 or 2.7 billion years ago, right? So they had to survive for a long time without having a lot of oxygen around, and probably they were doing it using a pathway very similar to what we see today for glycolysis. So another thing to think about is that glycolysis, as well as the citric acid cycle, connect to many other pathways in the cell. So we've been talking about these pathways as though they're kind of isolated events going on, and we look at these very simple cartoons that makes it look as though the cell is this big bag and it has just got these pathways going on and nothing else. But that's not true, at least from our discussion of the structure inside the cell. And in fact, these are pathways that connect up to lots of other types of metabolism that are going on, and not only to catabolic pathways in which we're breaking molecules down from more complex to simpler molecules, but also anabolic pathways where we're building more complicated molecules from simpler ones, right? And so anabolic pathways again would be things like generating starch molecules from individual units of glucose, or generating big proteins from amino acid components, or making RNA or DNA molecules from the individual nucleotides, right? And so the cell has to not only have energy just to do things like move around and move molecules around, it also has to be able to make molecules, right? So a lot of the energy that's generated in these pathways, as well as the intermediate molecules that are made, are actually feeding into these sorts of pathways that are building up more complex molecules in the cell. Oh, right, and I just wanted to also just mention that we talk a lot about glucose because we talk about glucose entering glycolysis, but you probably know that free glucose is actually not that common in our diet, right? Or in the diet of most animals. We're mostly getting our calories from things like fats or proteins, sucrose, other kinds of disaccharides, starches, and so these are molecules that have to be metabolized a bit first so that they can enter the pathways that we've been discussing. If it's in the case of starch, that's sort of easiest to imagine, right? Because those are long chains of glucose that are enzymatically cleaved off, and then we have individual glucose units that can directly enter glycolysis. But if we have proteins, let's say, that come into the body, then those have to be hydrolyzed down to the individual amino acid components and then further processed so that they can enter these metabolic pathways at one of the steps where there's an intermediate that can be made easily from, say, an amino acid being broken down. And so this just sort of summarizes some of this. So these catabolic pathways, again, these are referring to pathways that break down complex molecules to simpler ones. So these basically will funnel electrons from many different kinds of organic molecules that come in through the diet into cellular respiration. And importantly, glycolysis will accept a wide range of carbohydrates. Again, they have to eventually get in through being converted to glucose or to fructose or one of the other intermediates in the pathway, right? They can't be anything. They have to be in the pathway, but they can get in by being converted through other enzymes to those intermediate products, and they can just be funneled right into these central metabolic pathways. And as we said, proteins have to be digested first to individual amino acids, and these can then feed into glycolysis or into the citric acid cycle. Again, not as amino acids, of course, but as breakdown products from those that are then the same as some of the intermediates in those pathways. So a little bit more about that. So if we have fats, so remember that fats remember the structure of fats, right? We have a triacylglycerol, so we've got a 3-carbon molecule that has fatty acids that are esterified to each of those onto each of those carbons. And so these can be broken down. They can be digested to the fundamental glycerol unit, which is using glycolysis, so that can get into glycolysis. And then the individual fatty acids can actually be used to generate acetyl-CoA, so they can go right into the citric acid cycle through conversion to acetyl-CoA. And these fatty acid chains are actually broken down by a process called beta oxidation to yield acetyl-CoA. So one of the reasons that an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate is that we've got all of those very energy-rich carbon-carbon bonds in a fatty acid chain, right? Each of which can be used to generate multiple acetyl-CoA molecules that then go into the citric acid cycle. And your book shows a little diagram here that illustrates some of these pathways, and it's not showing you this in any kind of gory detail, but this is kind of the level that I want you to know this right now, is to just think about how these different types of molecules are able to get into metabolism at different steps. If we have carbohydrates, a lot of times we have simple sugars that can be broken down into glucose, and then they actually go into glycolysis as we've already discussed, right? So they enter here. But sometimes these sugars are other kinds of things, fructose, for example. So that would then enter glycolysis at a downstream step in the pathway. If we have proteins, these are first digested down to individual amino acids. Now, some of those amino acids might be used by the body to make other proteins, right? They might be funneled off. They wouldn't actually be fully oxidized through these sorts of pathways to generate energy. They would actually be used to make other proteins. But some of those amino acids could be further metabolized, as shown here, by releasing ammonia, so first getting rid of that, and then feeding the molecules that are then further metabolized by enzymes that allow them to either enter the pathway here as pyruvate or as acetyl-CoA, or sometimes downstream as intermediates that can get into the citric acid cycle. So it just depends on the amino acid and the types of enzymes that are around to understand at which step these breakdown products of proteins can enter metabolism. And then over here with fats, as we said, so these are typically going to be initially hydrolyzed to produce glycerol, which can get into the glycolytic pathway right here at the step of glyceraldehyde 3-phosphate. Fatty acids are these long chains that are then ultimately converted to acetyl-CoA. So they get in down here. They skip glycolysis. They are converted to acetyl-CoA, and then they get into the citric acid cycle right here. Yeah. No, it would actually be, she's asking how would it get into the pathway here. It would actually be converted to pyruvate through a set of enzymatic steps that would do that. And I wanted to just throw in a slide here that just points out that, and just to remind you to think about the fact that in addition to using these sorts of molecules to generate ATP, we're also using them to build other more complex molecules. So we have food molecules that are coming in. There's lots of other kinds of molecules that are in the cell. And if we have catabolic pathways are breaking these down. We're generating some ATP, sort of useful forms of energy here. We have some energy that's lost as heat, and then we also generate a lot of byproducts, and many of these are actually then fed back into various anabolic pathways that are used. These are, of course, energy-requiring pathways that use this energy that's generated in metabolism to build more complex molecules. So there's a really nice sort of cycle that's going on here constantly. So all of this is of course, very highly regulated. Now, when we talked about enzymes, we talked about the fact that, there's lots of ways to inhibit enzymes. And there's lots of situations in cells where that's a good thing, right, to have enzymes inhibited. We don't necessarily want everything cranking full bore all the time, right, because then things would be really out of whack. And so it turns out cells have very exquisite ways of controlling the flux through these sorts of metabolic pathways so that we only generate ATP when we need it. If we have a lot of ATP around, that tends to function to inhibit enzymes that are early in these metabolic pathways. And a very classic way that this works is through the process of feedback inhibition, which we talked about earlier, which, again, is just the idea that if we have an enzyme that's occurring early in the metabolic pathway that's sensitive to the product of that pathway, such as ATP, then it's a very beautiful way for cells to inhibit the flux through the pathway at an early step before a lot of energy is invested in the running of that pathway. So in metabolism overall, if ATP concentrations begin to fall, then respiration will increase. And conversely, if there's lots of ATP around, then respiration tends to slow down, right, because less ATP is required. And so the control of this is based primarily on regulating activities of these enzymes at strategic points in these processes, typically early on in the pathway. It doesn't do as much good to regulate the last enzyme in the process. It's better to do it early on. And I mentioned this previously, but I just want to point this out again because I think it's really important, and that is that one of the key enzymes that's regulated in metabolism is this enzyme called phosphofructokinase, which is shown here very schematically as the enzyme that's responsible for phosphorylating fructose 6-phosphate and converting it to fructose 1,6-bisphosphate. So it's the third step in the glycolytic pathway that we talked about. And why is this? Does anybody remember why is this particular enzyme really important in terms of regulation? Anybody remember? Oh, yeah. Right, so no, exactly. So he said it's basically it's one of the enzymes that's investing ATP in the cycle by phosphorylating fructose 6-phosphate and creating a molecule that's now essentially committed, right, to entering the rest of the glycolytic pathway. So it's really the important commitment step of that pathway. So cells highly regulate this enzyme. And I mentioned this previously, but there's been a lot of really interesting, beautiful, both structural biology with crystallography as well as biochemical studies on phosphofructokinase. And we know that this is an enzyme that is inhibited by ATP, and it's actually stimulated by ADP or AMP, right, the breakdown products of ATP hydrolysis. And it's also stimulated it's also inhibited when you have a lot of citric acid around. If you have a lot of citric acid around, then the TCA cycle is really cranking. Then we can inhibit PFK because we don't need to run glycolysis as fast. And the way this works is through allosteric regulation. So this is one of those great examples where we have an enzyme that's actually not a single polypeptide. It's actually 4 separate polypeptides that come together to make an active phosphofructokinase molecule. And within that tetramer are binding sites for AMP and ATP and citric acid. And depending on the concentrations of those molecules and the amount of binding that occurs on phosphofructokinase enzymes, the enzyme is either cranked up and really moving fast if there's lots of AMP around, for example, or it tends to be slowed down in the presence of a high concentration of citric acid or ATP. Right, so that's really important in terms of regulating all of this. And this is just sort of a cartoon that shows how these different products that are coming out of these pathways can trigger changes in the activity here that then, of course, alter the flux through the pathway here and then, of course, control what's happening downstream as well. And this is just a slide that I took out of an earlier lecture just to remind you of the actual chemistry that's going on here, where PFK is using an ATP molecule to phosphorylate fructose 6-phosphate, generating this diphosphorylated molecule that's now fully activated and ready to be split in half in the next step of the glycolytic pathway. So, this slide is just taken from, this is a figure from the textbook, and it just shows the kind of the overall picture of what's going on in all of these processes. So we have glycolysis that's going on in the cytosol, where glucose is being converted to 2 pyruvate molecules, and in the process, we're getting 2 NADH molecules made, and we're getting a net yield of 2 ATP molecules that are being made, right? And so down here, this is showing us the tally of ATP that's being generated in these pathways, and up here on the top, we're seeing the tally of these reduced coenzymes, NADH or FADH2, that are being made. So you know that, so once we have the ATP made here, that can actually be utilized for other sort of energy currency of the cell, right? Whereas NADH is a molecule that's actually a way of storing energy. It's a way of transferring electrons downstream to be driving oxidative phosphorylation through electron transport in chemiosmosis that leads to the production of more ATP molecules through that ATP synthase mechanism that we looked at last time. And so you should really make sure that you're familiar with this cartoon and that you know the yields of the different types of molecules that are being produced in each of these pathways, glycolysis, the citric acid cycle, and then ultimately oxidative phosphorylation. And overall, you know that we're getting the most bang for the buck from oxidative phosphorylation, right? That's where we're really making most of the ATP that gets generated. Okay, and then the last few slides here just summarize some of the things that we talked about previously. So I want you to really understand the way that this process is working. And the question that I asked you at the beginning of class about what happens if we uncouple the, if we were to break up the ability of the cell to generate this proton gradient, that's the type of question that you might see on the exam. So definitely think about, what happens if I inhibit these pathways at different steps? What does that do to the overall production of ATP? What does it do to the utilization of oxygen? And how does that affect the other functions that are going on in the cell? And so this is just a reminder that that ATP synthase is this engine that's sitting in the membrane of the mitochondria, and it's actually using this chemiosmotic gradient, proton gradient, to drive the production of ATP. And we saw last time how that kind of works, right? This thing is a really incredible molecular motor that uses protons to spin a turbine that generates ATP through conformational changes that activate the catalytic centers of this complex, right? So just really, I think, one of the most beautiful structures that I know of in biology. And then this is just a summary showing what happens in chemiosmosis, right? Where again, we've got the electron transport chain is going on here, protons are being pumped into this intermembrane space, and then being used to drive the production of ATP over here through ATP synthase molecules, and these ATPs are then generated back in the matrix of the mitochondria where the citric acid cycle is going on. And what the heck? Let's ask one more question. What do you think? One more iClicker, and then you can go have a nice holiday weekend. All right, so here it is. Yeah, just take a minute or so to do this. Okay, so about 15 seconds. Okay, 5 seconds. 4, 3, 2, 1, stop. Okay? Okay, very interesting. So let's see if we can, let me try to show you this. It's not letting me do it. Okay, so 50% of you said that the answer was A, and that's correct. So if you said A, good job, because basically these are cells that don't have, if they don't have functional mitochondria, they can't do anything beyond glycolysis. So they can use glucose, but they can't use any of those other molecules to feed into metabolism. All right? So if you said A, great job. Have a great weekend. See you Wednesday.